In this class, we will move on with the next topic, that is the formation of female gametophyte. Here, female gametophyte, it designate the embryo sac. Embryo sac. So we've already learned the structure of ovule. So let me just draw it roughly. Let me draw it. This is the chalazal. This is the micropyle. So this is the funicle. So here inside this ovule present a nucellar cells. So in our last class we have I have told you that one of the nucellar cell which is present towards the micropyle becomes bigger. So it becomes bigger, get bigger in size. So this one is the, let me draw it here. This one is the megaspores mother cells. It becomes the megaspores mother cells. The one which get bigger inside the ovule becomes the megaspores mother cell. So this one will get developed into a female gametophyte, that is the embryo sac. So this megaspores mother cells, it will undergo a meiotic division and then and it results into a tetrad. 1 2 3 4. This is the upper region, which is the chalazal. This one, and this is the micropyle. After meiotic division, it will form into a tetrad. This is known as a megaspores tetrad. Megaspore tetrad. Megaspore tetrad. This megaspore tetrad, after some time, only one cells it survive. Only one cell it survive. So this is the only one cell survive. So I will draw with these three cells, it get degenerated. And the one which is on the chalazal, to the three cells which is towards the chalazal, they got degenerated, and the one which is towards the micropyle is the survival megaspores. So this one will generate, and this is the survival megaspores. So this survival megaspores. So this is the, we always say the upper region is the chalazal and the lower is the micropyle. So these survival megaspore cells, it will undergo this survival megaspores, it will undergo 3 mitotic division. 3 mitotic division or 3 karyokinesis. Karyokinesis. So this is the chalazal, this is the micropyle. So first mitotic division, it will give 2. Second, it will give 4. And third, it will give 1 mitotic division, it will results into 2. So that is the first mitotic division. Second mitotic division, from 2 cells it will results into 4. So into 4. And then in the third mitotic division, from 4 cells it will results into 8. So that is the third mitotic division. And from here, the cells move by itself. Suppose three cells will go up, 3 cells will go up, 3 will go down, and 2 will stay at the center. These 3 cells which is present towards the chalazal is known as the antipodal. And then these 3 cells which is present towards the micropyle, the cells which is in the middle, it is known as the egg. And the 2 other cells which present on the side, on each side of the cell, is known as the synergid. And these 2 form the egg apparatus. And the 2 nucleus and the 2 cells which is present in the center, these are known as the polar nuclei. So now we shall count how this. So this is a fully developed nucellar cells. This is a fully developed nucellar cells which results into a fully developed embryo sac. So this embryo sac, it contains how many nucleus? We shall count 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated. And how many cells? How many cells? It is made up of 8 nucleated. And how many cells? This is 1 cells, 2, 3, 4, 5, 6, 6, and then this whole cell, which is made up of 7. So 7 cell. This is known as a polygonal. This is known as a polygonal type of embryo. Polygonal type of embryo sac. A polygonal type of embryo sac is very common in angiosperm, and it is developed from a monosporic that is from one nucellar cells, and it is 8 nucleated and 7 cell. It is known as a polygonal type. This embryo sac is known as a polygonal type. So that concludes the formation of a female gametophyte, which is the embryo sacs. And in the next class, we shall continue with the types of ovule, since there are many types of ovule in an angiosperm.